I'm Sebastian. This is a continuation of the previous video where we looked elaborately at the structure and the functions of mitochondria. Now, in this particular video, we will also look at how proteins are transported. The proteins which are synthesized in the cytoplasm, how they are transported into the mitochondria. Now, in mitochondria, there are 4 locations to which proteins must be translocated. The first one is the outer mitochondrial membrane, then you have an inner membrane space, then you have the inner membrane, then finally the mitochondrial matrix. How very specifically proteins that are synthesized in the cytoplasm are targeted towards this particular place? That is going to be the point of discussion at this moment. And this is carried out by something called a translocational proteins, or it's a translocational motor. So we will be elaborating on the mechanism of translocational motors. Scientists have been wondering how this protein which are synthesized in the cytoplasm is transported across the membrane. Blobel came up with a hypothesis known as the signal hypothesis. Signal hypothesis was originally formulated for transport of proteins to endoplasmic reticulum. We will look at it later on. But in this signal hypothesis, what it says is every protein will have a signal. And that signal is present normally at the N terminus. This signal directs the protein to a particular location. Scientists wanted to prove it. It is the signal which will direct the protein to a particular location. So they carried out very simple experiment. And one of the aim of explaining this experiment is that we get enlightened how these the theories are proved. So what is done is simple experiments like they took a protein. Assume this is a protein. This is a protein that has to be inserted into a particular location. And this protein will have a signal. So this is the signal. The red one is the signal. And this is the protein to be targeted to the mitochondria. Now this is taken in a centrifuge tube, in a test tube. Now, if the first experiment which was carried out is, this is treated with the trypsin. You know that when it is treated with the trypsin will degrade the protein. So if the trypsin degraded the protein into small fragments, okay? If the signal also got degraded into small fragments, so it is there in the test tube. You can prove it either by a protein estimation or by a page. SDS page will be able to help us to identify these proteins. A very simple experiment. Now, they also carried out another experiment. What they did in the next experiment is, now this is treated with the mitochondria. Mitochondria is added. Respiring mitochondria. So, respiring mitochondria is highly active. And this is taken from yeast. So, yeast respiring mitochondria is added to this. So, according to our theory, if there is a signal which is meant for mitochondria, this protein must get inside the mitochondria. So, what was observed is, assume this is the mitochondria, this is the inner membrane. What was observed is, they found that if the protein got inside the mitochondria and if the signal is here, signal, we'll put a red so that no confusion, signal is also there. Protein got incorporated and this is now in our test tube. So in the medium there will not be any protein. You will not be able to determine protein from this. Okay, because the protein got incorporated into the mitochondria. Now what was done? Treat it with trypsin. When you treat it with the trypsin, what should happen? Proteins must get degraded. But there is no protein in the medium. So, the experiment will prove that you have the mitochondria which is intact. Okay, the mitochondria is intact and you have the protein inside and nothing happened. Trypsin had no effect. When you carry out a page, you will get no band at all. So this was a very simple experiment carried out in order to prove the importance of the signal. And now look at the signal. So they began decoding the signal. Now what was realized about the signal is normally a short stretch of amino acid that is roughly about 20 to 50 amino acid. That is a signal normally present at the N terminus, and that is heavily of hydrophobic amino acid. Hydrophobic amino acid, and it also has positively charged amino acid like arginine, lysine, etc. and no acidic amino acids present in this. This was an observation made. And this signal normally forms a kind of an alpha helical structure. So in the alpha helical structure, again what was the observation? If the hydrophobic amino acids are at one side and the positively charged amino acids are at the other end. This is significant. Because, why it is significant? One side becomes a kind of an amphipathic. And this signal will bind to the receptor based on this hydrophobicity or based on the charge, the positive charge. So this is the general nature of the signal which they have identified. Now the question is, how exactly this translocational motor operates, or how is the protein which is present in the cytoplasm is brought inside. So in order to understand that, we need to look at the complexes that are present on the membrane. So if you consider, this is the outer membrane and this is the inner mitochondrial membrane and this is the inner mitochondrial membrane space, and this is roughly about 20 to 30 nanometer is the size of this inner membrane space. Now on the outer membrane there is a complex, a protein complex, and this protein complex we shall call it the TOM complex. TOM stands for translocon outer membrane. Translocon outer membrane. It's a complex. There are many proteins associated with this. Two important proteins which are present here. One is the TOM20. It should be based on the S value. And another one is the TOM22. These two are forming in the kind of a receptor for identification of the signal. Then you have another TOM protein that is called TOM40. TOM40 becomes a channel. So here is a channel. This is a real channel which will allow the protein to be transported inside. So what generally happens is, you have a protein here. Okay, you have a protein, and this protein is associated with the molecular chaperone. There will be heat shock proteins. So let us put, this is the signal, the red one is the signal, and it will have heat shock protein, and heat shock protein we shall write HSP, and the heat shock proteins are 70, and they will be associated with this. Okay, and the first thing is to remove the heat shock protein. In order to remove the heat shock proteins, what is needed is an ATP hydrolysis. So ATP hydrolysis occurs. ATP is broken down to ADP plus PI. That energy is used in order to separate these heat shock proteins. So heat shock proteins, once it is separated, the chaperones are separated, then this protein becomes a linear molecule. So there is a very interesting experiment they carried out in order to prove that the protein must be linear. Okay, it has to be unfolded. And that experiment, let me explain here that experiment. So what they have done is, there is a protein meant for mitochondrial matrix, that is the alcohol dehydrogenase. We know that alcohol dehydrogenase will have the signal meant for the mitochondrial matrix. There is another protein which is supposed to be in the cytoplasm, and that is dihydrofolate reductase enzyme. It is meant for cytoplasm. So by all means, DHFR should not be transported into the mitochondrial matrix because it does not have the signal. What scientists have done is they have taken the signal from here and attached to DHFR. What will happen? DHFR, because of the signal, will be recognized by the Tom complex and it will be transported into the mitochondrial matrix. And they proved it. They said, yes, it is happening. And they could see the presence of DHFR in the mitochondrial matrix because of the signal. They did another experiment. And therefore they proved the role of the signal. Another experiment was carried out. They wanted to check, has to be unfolded in order to bring it inside. There is a medicine which is used for treatment of cancer, that is methotrexate. Methotrexate is a medicine which is used against cancer. What methotrexate does is it will bind with the DHFR and it inhibits the enzyme. It will not allow the protein to unfold. So DHFR will remain in the folded manner if methotrexate is added. So they added methotrexate to it. So if you consider this is the Tom complex which has to transport the protein, now through the channel, signal will identify if the signal comes inside, but if the methotrexate bound DHFR, this is a methotrexate bound DHFR which is not unfolded, stays back in the cytoplasm. And that is how they proved that the protein must be unfolded. And they also did another experiment to prove that energy is needed for bringing the protein here. So here we are talking about ATP. And energy comes in two ways. One as ATP molecules, another one in the form of a proton motive force. We are well aware that in the electron transport chain, which is present in the inner membrane, it will transport a lot of protons to the inner membrane space. And this positive charge is essential for bringing the protein to the mitochondrial matrix. So energy in the form of PMF is needed. So this can be very easily carried out. We are aware of the inhibitors of this electron transport chain. They have used DNP and cyanide. DNP, cyanide, etc. are used, inhibited and dissipated the charge, and to prove that this PMF is essential for bringing a protein inside the mitochondrial matrix. So two things are proved. One, it has to be unfolded, and the second, energy is needed in the form of a proton motive force. They proved a third point. The third point which they proved is in an experiment is, if you look at I have rubbed the inner mitochondrial, the TIM complex, that is a translocon inner mitochondrial membrane. TIM and the TOM are closely associated, and they are very close in the mitochondrial membrane. Wherever the protein has to be transported, you will find them closely related, closely placed. So if the protein which is going to pass through has to directly, it will go to the TIM complex. If they are not close by, if the protein finding it difficult to reach the mitochondrial matrix. So what they did, they put a spacer here. So this is a spacer, a set of amino acids. When the spacer is about 50 amino acid, this is able to come to the inner mitochondrial membrane. When the spacer is only about 35 amino acid, it cannot reach the TIM complex. So therefore, if the protein is not able to be transported into the mitochondrial matrix. Look at the beauty of these experiments. 3 experiments we have seen by making the protein unfolded, making the protein with a short spacer, and dissipating the PMF. They proved a lot of points. See, look at the brilliance involved in carrying out these simple experiments and what kind of conclusions have been drawn from this. We still have not explained the process of transporting these proteins. Keep in mind these 3 experiments that were carried out, and now let us see how this protein is brought inside. We are back to our original picture. So this was our original picture. So this is the outer mitochondrial membrane, this is the inner mitochondrial membrane. Tom has Tom 20 and 22, which will act as a kind of a receptor, identify the signal. Once it identifies the signal, this is Tom 40, the green one, that will open. It will open and it will allow this protein to come, okay, and the blue is the protein and the protein is pulled. One important aspect we should not forget, especially with regard to the identification of the signal peptide. I skipped that, I am repeating. That is, we have seen that in the alpha helical signal, one portion is hydrophobic. Another portion is, it is with a positively charged amino acid. What this Tom 20 and 22 does is, to Tom 20 will recognize the hydrophobic amino acids. It forms a kind of a connection. But this linkage is very weak. Now it has to be strengthened. And that strengthening process is carried out by Tom 22, which has got acidic amino acid. And those acidic amino acids will react with these basic amino acids, and now the bonding becomes tight. So if the signal is properly identified. Now with that, this signal will be channeled. Now this channel opens, that is a Tom 40 opens, and it will allow the protein to pass through. So this is how, if the Tom allows, if the protein destined for or meant for, if the mitochondrial matrix is identified. Once this occurs, now look at the next complex. If the next complex is the TIM complex. TIM stands for Translocon Inner Mitochondrial Membrane. This is also a complex. And this complex has, especially you have TIM 23, TIM 17, this becomes a channel. Then you have also TIM 50. TIM 50 identifies. After TIM 50 identifies, it brings in a structural change, a conformational change in TIM 23. And this conformational change will allow this particular protein to pass through. So therefore, you can see that the signal comes here, the signal is routed here, and the protein is allowed to pass through. Okay, now also remember, as we have seen earlier, this Tim and the Tom complexes are in such a place where if the distance is very minimum, there will be almost direct interaction. Directly this protein is allowed to get into the Tim complex so that it is not lost in the inner membrane space. Directly it is transported to the Tim complex. Another important aspect is about as the protein is being pushed inside. Now it is like a tug of war. Not exactly, sorry, it's not a tug of war. It is like pulling a rope. It is like pulling a rope towards you. What happens is, inside the mitochondria, you have the HSP proteins, HSP70. Now the HSP70 will bind on this. If the HSP70 will bind on this, and through hydrolysis of ATP molecules, ATP hydrolyzes to ADP and PI hydrolyzes, and that energy is used for pulling in this blue protein into the mitochondrial matrix. See how the translocation occurs. Acting as a motor, it is translocating. By pulling, it brings the protein into the mitochondrial matrix. ATPs are used at two places, one in the cytoplasm, another one in the mitochondrial matrix. And the second energy, which we have seen earlier, that energy is the proton motive force. The proton motive force which exists across this inner mitochondrial membrane, that also helps to bring the protein inside the mitochondrial matrix. Two forms of energy. Once the protein is inside the mitochondrial matrix, now we come to the last phase of it. That is how this protein is folded and located at the right place.